Hello and welcome to the Sound of Science. Today I want to speak about the differences between a traditional drug development and the SARS-2 vaccine development was highly accelerated, and this is based on a paper recently published in the journal Nature on September 2020. And therefore, on traditional development of drugs, there is an initial preclinical stage which includes the study design and conducting of early experiments that takes for several years. However, in the case of SARS-2, it lasted for several months only, and one of the most critical reasons for that is the vast experience that we've gained after the outbreaks of the SARS-1 virus in November 2002 and the MERS outbreak, the Middle East Respiratory Syndrome outbreak that occurred on April 2012. And all of that really prepared us well for the SARS-2 outbreak and on December 2019. And so following these early preclinical stages that typically again last for years but lasted only several months in this case, there is a process development and toxicology assessment which aims at understanding whether the vaccine or drug are toxic and at what doses to the organism, as well as starting to think about how to implement the process of commercializing the product. This is to a preclinical stage that typically lasts for years. However, in the case of the SARS-2 vaccine development, only lasted for several months. All the data gathered from this stage are sent as an investigational new drug to the FDA or to the EMA. And following approval of this investigational new drug, there is a phase of clinical trials, which is comprised of phase 1, 2, and 3. Phase 1 aims at assessing safety of the drug. It typically lasts between 1 and 2 years, and it involves less than 100 individuals. Phase 2 aims at understanding immunogenicity and the relevant doses that the drug or the vaccine should be administered at. It typically lasts for 2 years and it involves few hundred individuals. Phase 3 aims at assessing efficacy and safety of the treatment. It is a relatively long phase. It lasts for 2 to 3 years and it involves thousands of individuals. Now here is a major difference between the traditional drug development and the SARS-2 vaccine development. You can see that the phase 1 and phase 2 clinical trials were actually staggered and overlapping. And following the completion of these phases, there was a rapid assessment of the outcomes of these phase 1 and 2 clinical trials, and phase 3 was immediately conducted. And so phases 1, 2, and 3 of the SARS-CoV-19 vaccine development only lasted for several months. However, these phases were not compromised by the number of individuals, for example, that participated. For example, in the Pfizer phase 3 clinical study, there were over 40,000 individuals participating. So following completion of phase 3 clinical trial, there is a biologics license application that is being submitted to the relevant regulatory body, for example, the FDA or the EMA, and it is being assessed in the regulatory body for between 1 and 2 years. However, this is another major difference between the traditional development and the SARS-2 vaccine development. It was very rapid assessment by the FDA. A lot of bureaucracy was cut away, and assessment was very rapid. Another big difference between traditional development of drugs and the SARS-2 vaccine development was that during phase 3 of the clinical trial, the companies began production of the vaccine at risk, which means that in case that the phase 3 clinical trial would fail, or that the relevant authorities would not okay this vaccine, this was a very big risk that they took upon themselves. A lot of money was invested here. Whereas typically, following completion of phase 3, there is a submission of the BLA to the regulatory board, for example, the FDA, and only following completion of the review by the FDA, only then there is a large-scale production, typically. However, this was not the case, and the pharmaceutical companies took a big risk on themselves because there is a lot of money involved in here. And so if to summarize, traditional drug development lasts for more than 15 years, typically. And one of the reasons for this lengthy process is that every step there is an economic risk assessment during this process, whereas the SARS-2 vaccine development did not have that risk assessment, and therefore most vaccines were developed within 10 months to 1.5 years. It is important to understand that moving forward was enabled at a financial risk by the pharmaceutical companies, and this is what enabled the accelerated development of vaccines for SARS-2 virus infection. And so with that, I would like to conclude. If there are any questions, I will be happy to answer them in the comments, and until the next time, stay safe.